12 to noon, and then the second time I did it was at night. And then the advanced lecture was backwards. So yesterday I was like, at 8 o'clock. And by 10 o'clock, I was like, by 10 o'clock, we were going into this life session thing, right? Like, let's have this sort of freewheeling chat. And I was like, okay, I should remember to be positive because, you're tired and all the cynicism comes out. So I tried to be positive and upbeat. But then if you meet anyone from the night shift and they're feeling really depressed after talking to me, that's why. All right, so let me find my lecture. I will share it with you and play it. All right, so thank you all for joining me again. This is part 2 or the advanced portion of the festivities. So I had this, crossroads kind of thing, right? Like we could do a much more in-depth dive into some of the mathematical foundations and like that. Or which would be much more fun if we were sort of meeting in real life, and there was a board and we could take turns working together on the board and like that. But otherwise, just slapping a bunch of equations on here, just didn't feel like I would do the right kind of service to this course. So what I thought instead was, under the umbrella of what we can learn about the brain from RNNs, I would take that literally and show you a few applications of recurrent neural networks for various purposes in neuroscience. I will touch upon a few popular examples that you may have encountered. I will do a in-depth dive into one of the current projects in my lab, so you can see how quickly you can ramp up from the kinds of foundations that we learned in the first, in the basic lectures, to something that is essentially pushing the envelope sort of experiment theory collaboration and a research project. So, I'm trying to do the opposite of the spirit of the advanced lectures, not to sort of intimidate and drive away all of you, but to rather say that all of these problems, even at the very advanced level, are tractable. If we simplify the deck, sort of in our training as physical scientists, to clear the deck, pick a small sliver of the problem, get comfortable with some of the assumptions, and see how we can solve some of those problems. So, in the basic lecture, I thought that I was trying to impart to you this idea of the elegance of recurrent neural network models, particularly in the simplified cases, and the advantages of using something that had the dynamical range to produce interesting sorts of time-varying behaviors, but did not come at the cost of something super trivial either. And mathematical tractability came along for the ride. So today I'm going to talk about what it means to train recurrent neural network models, or indeed any type of neural network models, to do something. So what do these terms mean when you hear people saying, oh, I trained this network? Is it really curve fitting, as is commonly disparagingly said, or is it something slightly more than that? What does it mean when you reverse engineer RNNs that have been fit to time varying data? What types of analyses could you make use of? Where this type of approach has been helpful for us in neuroscience? And maybe if there's time, collectively, we can jot down a few things where this type of approach can be taken in the future. But the last part is speculative, and we can see how we do in terms of time. All right, so what I want to do is to give you this idea of dynamical motifs. So let me begin from where we left off in the previous lecture, which is we together developed this idea of randomly connected nonlinear RNNs. We saw those types of networks, and here's the activity of a single neuron in one of those RNNs. There's random connectivity. It is still wildly unrealistic. It has no external drive. In the thermodynamic limit, networks like this, I told you, look chaotic. So I'm picking a randomly picked neuron from one such simulation as a function of time, and it's irregular. We discussed what happens when inputs above a certain critical amplitude go into networks like this. We discussed certain properties of this phase transition that let these types of networks quench their ongoing intrinsic variability and start to oscillate or get entrained to the frequency of the input. We also saw what happens when, I'm just showing you one neuron, but even at the population level, this type of switch from spontaneous ongoing dynamics, which are correlated over multiple scales, become entrained to the frequency of the input, and you see something much more regular from these types of networks. So one way of improving the reliability of these networks is to give them inputs. Now, okay, I can show you these type of population plots, and indeed experimental papers are also sometimes filled with this type of look at my pretty picture kind of plots. But one way to succinctly make sense of population level data like this can be something that, you know, theorists, computational neuroscientists have come up with, and that is the idea of state spaces. So state spaces are nothing but a coordinate frame that you can derive by applying dimensionality reduction techniques to population level data. So this is, you know, an N neurons over time. That entire population can be succinctly described in this coordinate axis. And if you did some kind of dimensionality reduction on this, you can do GPFA, you can do PCA. There's a variety of this. In fact, there's a whole industry of computational neuroscientists developing dimensionality reduction techniques. But let's say I just stuck with PCA for the purposes of today to keep things simple, as is our mantra. When you do that, you find that spontaneous rich activity like this traverses some kind of trajectory here in state space, right? So take, for example, this state space trajectory. Once this network is entrained to the frequency of the input, some of this external variability goes away, and you see the same network now exhibiting cyclical or oscillatory activity corresponding to the sinusoid, the frequency of the sinusoid that was applied to it. So this notion is essentially leading us to start to develop the concept of the geometry of these types of dynamical motifs. So this is the idea of dynamical motifs that I'm introducing. What do these patterns look like in state space? So we're you know, together in the first lecture, we tried to write down a theory of cognition as dynamics. And yes, it sounds kind of trite, but really it's deep in that anything we do in terms of learning, remembering, behaving, deciding, all of those activity patterns, all the activity patterns responsible for those behaviors. And indeed, those behaviors themselves are time varying. So to my eyes, and it's a taste in problems thing again, at least to my eyes, talking about cognition in the absence of dynamics is not a sensible way to proceed. So we tried to write down some of the pieces that came with this idea. Now we want to enrich this theory of cognition as dynamics with the concept of geometry of the dynamic motifs and state spaces. So that's what we're going to do here. And we want to see how those types of concepts will help us simplify some of our understanding of neuroscientific phenomena. So back to our, you know, famous drawing board, we started building a theory of cognition as dynamics, and we looked at what happens to neural networks when you start to give them periodic inputs. So here's an equation for the driven nonlinear RNN. X dot is a simplified way of writing dx dt is minus x plus the current J phi, which is the recurrent currents from everyone. And I don't mean to be pedantic here. This bears repeating because this J phi term becomes critical to us a little later when we try to look at state space projections of various quantities here. Plot this external drive. So without any inputs, we got kind of trivial fixed points or activity decaying to zero, or we got untamed chaos, which had repeatability and reliability problems. So we put inputs onto them, and because of this phase transition that we looked at, we found that we got kind of the best of both worlds. We got regularity from this this lava lamp, but nonetheless not you know, not super interesting behaviors, right? So then the question is, are we done? The answer is no, because the brain does much more than reconcile subtle inputs and ongoing activity. So you want more from this lava lamp than just blind reading of the patterns that were played to it from the external environment in the first place. So you want more than a blind pattern generator, right? So you want behaviors that at least remind you of some of the richness of Heidi the octopus having a dream over time. So in order to do that, what we're going to do in this lecture is to start to develop away from randomly connected J matrices. What did the randomly connected matrix buy us before, right? It bought us the ability to say something general and understand something fundamental about properties of large networks with as few assumptions in the connectivity as possible. Now, sacrificing that statement of generality will now help us to do something interesting with these networks, right? So that's why we want to move away from what I've called JRAND here and start to train networks to do something. So when I say, or when anyone in my field says, we trained this network to do blah, what they mean is that they've changed the weight matrix or the recurrent interactions within the network typically to something else that is much more structured, therefore much more specialized for the thing you want to do, and therefore away from the general kind of statements and theorems that we were able to discuss in the random case. But nonetheless, we're able to, by means of training, do something useful for these networks. So what we will see today is how this approach has been applied profitably to get networks to flexibly perform different tasks. And when I say tasks, I just mean in the experimental setting, but people are developing RNNs that can be trained to generate more naturalistic behaviors as well. So for today's talk, let's just keep the definition of task kind of loose. Now, what we want these networks to be able to do is to solve the problem that the JRandom network had, which is to have reliable and repeatable performance. And once we do that, we also want to be able to produce those behaviors from dynamics that are reliable and repeatable, but still not trivial. Now, by doing this, we want to be able to discover mechanisms. So I'm now using the dreaded M word. I'm also going to use another dreaded M word a little later. But what do I mean by mechanism here is just, again, a colloquial definition of getting at some kind of quantities that you cannot get at from measurements alone. So I'm not putting a stringent causality condition or anything like that here. I'm just saying, can we get to some understanding of the biological system beyond the details of a single experiment or idiosyncrasy specific to a certain species or a certain instantiation? That's what I mean by mechanism discovery. Okay, so how do we do this, right? So here's a little brief kind of, lightning speed survey into the state of the field. So there are many approaches for building and training recurrent neural network models in neuroscience. And here are a few key references. I am particularly fond of Omri Barak's review on the subject, but there's several excellent ones. This list is by no means exhaustive. And as I keep going, you will see many references like this appear. I will make the slides and this reading list available to you after the fact, once I figure out how to share Dropbox link by email to Schwan probably. But anyway, back to the science of it. So the normative way of doing something, and I will define in a second what that means, is if I want a network to do something, the best approach would be for me to imagine how this task should be performed. The optimal way of performing a task, and then I want to dial that mechanism up in the circuitry of the network, right? That's the best way to do it. Now, this is possible and has been done by several clever people. And you can understand by this whether or not the biological system used the same mechanism or not, right? That said, there's only a few problems for which this approach is really nicely solvable. And I'm giving you a few examples here. I wish that I were clever enough to come up with these at all, but I haven't been so far. So for example, if you want to do a task like, let's say you're standing in front of the elevators, right? The elevator doors open, the lifts, for those of you who are in Europe, I suppose. So, let's say 2 doors open and, people are getting into the doors and you're just standing there trying to figure out which side to get into. So this is almost an integration task, right? Because evidence is appearing as people keep getting added to this elevator bank. You are trying to decide on a moment by moment basis which side has fewer people and get into that elevator, right? This is let's say there's 2 channels going and you're just counting pulsatile evidence. There's a whole class of networks that have been built for evidence accumulation. There's a whole class of working memory tasks and decision-making tasks that rely on this. Now, we know how to do integration, right? Integration could be done by means of fixed points. So you could imagine that in the state space of my brain, somebody is accumulating a set of fixed points, one for each channel, let's say, that is integrating something, right? And once something is integrated to a bound, I make a decision and go into the elevator, which has either least people or more people, depending on your tolerance for risk, right? Now, let's say that this task weren't taking place in the elevator, but really involved the computation of me going from my apartment all the way to the elevator that's at the end of the hallway, while determining that every person getting into the elevator was also dinging the elevator in some way. So this means that auditory signals, which are sometimes transformed into log scale in my brain, have to get integrated, right? So we don't know how the naturalistic behavior is getting encoded. I can't imagine a mechanism for something like this. So I don't know what to look for in state space, right? If I know a priori that I'm doing an integration task only in linear units, I can look for a fixed point in state space. And again, there's a cottage industry for doing something like this. But when you move away from something so stereotypical, this ability to imagine a mechanism and dial up into the circuitry tends to fall apart a little bit because it's limited by our own imagination, right? Okay, so then came explosive advances in machine learning, computer science, and some by computational neuroscientists and theorists amongst us. And that involved training models to do things. So that involved taking the task and training a network to do this task. So you can imagine an artificial agent that has to do this navigation task of leaving my apartment, going to the elevator and getting into whichever block you want. You can train a network model to do this, right? To respond to a string of pulsatile inputs and then pick one. And those types of approaches became extremely popular in order to understand mechanisms. And in that category of training networks to do something behavioral is models that involve force, backprop through time, gradient-based training methods, and so on. And we'll touch on some of those. And I've done this myself. Then there's a third class of using network models to infer mechanisms. And that involves using network models as hypothesis generators, or using network models that have been trained to match empirical data directly, and then using those as a test bed to either falsify or validate mechanisms. And that's what connects the middle bar to the training procedure. So the network uses opportunistically, it constructs a solution, but one that is constrained by the details of the experiment or data collected in naturalistic setting. And so, I have done most of my work in the second two categories, but here are people whose names you might wanna, vaguely remember, so you can go back and look at some of these papers, and some very beautiful work has taken place in all of these. Right? So that's the kind of stuff we're going to be talking about today, is the approaches for building and training neural network models. Now, let's look at what something like that would entail, right? So training algorithms for recurrent neural networks to perform something brain-like. This equation is something we've seen 100 times before, at least, right? It's the nonlinear RNN equation. X dot is minus X plus J phi, as you saw like 2 slides ago also. So this is just the solution to this equation is describing over time how the current in neuron I would behave, right? You know everything you need to know about a model like this if you knew everybody's firing rates, which are the phi's, and how strongly they're coupled to one another, which come out in this matrix J. Now, if I want to take a network and train it to do something, I might as well attach a readout to it. And that's another common approach, which is to take all the phi i's from within the activations of this quote unquote reservoir and using a weighted readout that can produce an output, right? Like an output unit. So you can imagine that the entire rat or the mouse is in the schematic on the left, and the rat or the mouse's snout is this output unit, right? Because that's where the mouse is heading or turning to make a decision. Right? That would be the output unit. Now, if this network is connected randomly, everybody is chaotic, the output unit would produce an output that is also irregular. Now, the equation for the output unit's activation is just the weighted sum, as I told you, of everybody else's activations, and the readout weights can be made plastic. So you can say, whatever processing is occurring in my brain, what are the weight transforms that need to happen for the mouse to move its snout to the right or the left, depending on which elevator it wants to get into, right? In addition, the recurrent weights inside the brain of the rat or the mouse, or in this case, our randomly connected network, randomly initialized network, can also be made plastic. And that's what learning algorithms do. They can either change W, or they can change J, or they can change both, depending on what kind of constraints you want to apply to such a thing. Now, what we're going to be looking at is what happens at the end of this, right? At the end of this, what you want is a specialized J. You want a J matrix that has the dynamic complexity that you're looking for, that you had from the random J already, but does not suffer from the issues of the random J in terms of the reliability and repeatability issues, or that every time you run this network, you don't have some randomly oscillating mouse getting into random elevator banks everywhere. You want this mouse to get into the elevator you tell it to get into, right? Trial after trial, you want repeatable performance. So what you want to do is to move from J random to J trained. Now, in terms of learning algorithms, and for our younger colleagues here, those of you who don't do this for a living, I'm essentially blasting past entire semesters worth of stuff here, right? So if you're feeling a sense of panic, that's normal, let it pass. And all of this then becomes clear with enough repetition. So when you look at training algorithms that actually take you from this random J to a something task specialized J, there are essentially two dichotomous categories that I can think of. So if I wanted, when I wanted to make this lecture, I thought, well, should I give them like a whole list of possible training algorithms that are popular in literature? And I thought that that would be kind of messy. So I looked for the most dichotomous examples that I could think of, and I picked one from each camp. So I think of sort of reward-based learning mechanisms or behavior level models to be trained by using gradient-based methods, and there's target-based methods. And let me get into a little bit of detail about all of them, right? So these are the two categories. There's many other possibles. Within these categories, there are many examples possible. So you can pick your poison, but I can justify why I'm picking these two. So in gradient-based methods, here's our same famous, rat with the snout schematic, right? You've put an output unit onto the reservoir. Initially, all the weights in the matrix J are still random. There's a readout unit, which produces the activity over time Z, which is the weighted sum of everyone else's activations. And our goal here is to train the recurrent weights, which are initialized randomly, to go towards something. So the first thing we want to do here is to figure out what you want the rat to do or the mouse to do, or for me to do when I'm standing in front of the elevator. So I want to define myself an error function, which is where am I, where is my head pointed right now, and where it needs to be at some time. That would be the error. In gradient-based methods, you're optimizing the J matrix by simply the gradient of the error. That's what this equation is telling you. There's a learning rate that tells you how frequently you want to do this, but dE over dJij is simply the gradient of this error that you've defined. I gave you an example error, right? My instantaneous position minus where I want to be, but you can define many other such errors, right? Like, for example, propagate this weight only if I get into the right elevator and somebody tells me you got into the right elevator, right? And so there's varieties of putting more complexity on this, but gradient-based methods literally optimize the matrix based on the gradient of the error that you define. The error is typically defined only on the output and is propagated into the network. So there is one error that's defined on the output. That's the key to remember here. There's many papers. Again, I've put a few labs of the people whose papers you will encounter in literature that have published extensively on this. Then there's a class of methods called target-based methods. Sometimes they're called teacher forcing methods. We stole this again from the machine learning crowd. But for target-based methods, the update looks a whole lot like the one in the left, except the update term is the linear error between everyone's activation and a target. So the first thing you want to do for a target-based method is maybe you don't need the output unit. If I have data for everyone in my brain from a rat that's been trained to do this task over millions of trials, then every unit can just have its own target. And that's what phi I minus fi does. The error is now the instantaneous linear difference between where every neuron's activation is and where it needs to be. And there's a multiplicative regularizer, which I can get into the details of in a second. Where do these targets come from? These targets can come from actually data, which I have done myself. You can literally take experimental data and try to ask the question, what do you need to change in the connectivity of a network in order to autonomously produce time-varying activity like this? Once training is done, the network should stably produce the full dynamical system. So you get the covariance of the activity for free, but you also get stability of the activity for free, right? Otherwise, you could have just fit the covariance of the data. You don't need a theorist for this. This trains the full dynamical system. And this is why this approach is not exactly curve fitting. If I fit a curve to a static picture of a covariance, in the next time step, if the covariance is changed, you're shit out of luck. This thing captures that whole pattern plus all the off manifold directions. We'll again touch on some of these advantages as we go along. But if you have targets for all the activations, you can use them to train your network. Now, the multiplicative regularizer is a function of the average autocorrelation. So what does that mean, right? This P matrix that you see here, that means if I'm the ith neuron, and let's say there's 5 guys connected to me upstream, everybody fires one spike, right? Without this P matrix, I want to strengthen all my synapses 5-fold, right? You see why that would be? Because everyone else is firing 5-fold, they're connected to me. So I'm going to strengthen all of my synapses 5-fold. And that will blow up the activity in the network, right? So it can cause these pockets of instability if the error was just the linear difference between your activation and where you need to be. What this regularizer does is, in sort of this artificially contrived way, as are all these learning rules, balance the covariance of activity throughout the network. So if everybody ahead of me has fired 5-fold, I only strengthen my synapses a 5th as much. So I don't blow up, in other words. So let's say you don't have data for everybody, right? So there's obvious problems with this learning rule, right? Like, for example, where did the targets come from in the first place in the brain? We don't know. Mike Halassa thinks that maybe the thalamus produces some target functions for the cortex to learn, but we don't know. It's tenuous. Let's say you had a sparsely sampled population, like you're working with non-human primate data. Then what does it mean to train a network to produce that kind of pseudo population, which could be trial average, which could be from multiple sessions, which could be from multiple monkeys, and so on. So there's issues with this, just as there are issues with all learning rules used for training networks. But let's say you didn't have targets at all. You can go to the old faithful teacher forcing case. Right? You can use the output of another network. Let's say I know how to use normative models to do integration. So the normative model produced by one of the normative model labs could be used to produce target functions that I can use as constraint to train my favorite network, and then see what type of connectivity could cause that type of model to still be true. So target-based methods and gradient-based methods, there are 2 different sort of camps. We're currently working on a manuscript to show that they're not as dichotomous as we think they are, that in some domains, the gradients and this linear difference could look similar. And there are some advantages to the multiplicative regularizer, which means that gradient-based methods should also probably consider using such a multiplicative regularizer. But you guys can wait for the ICLR paper for something like that. But here are a few more labs that have worked on target-based methods to train these networks. Right. So before I dive into details of what happens when you do this, what does it mean by mechanism, I wanted to highlight my 3 favorite pieces from literature. For no reason except that they're the most recent in my mind, and 2 of them have rock star women scientists behind them. One of among many others, right? So there's literally thousands of papers now that have used RNNs in neuroscience. So here's arguably the most famous one, right? This is Valerio Monti and David Susillo's paper, where they've taken an RNN, and I'm only going to touch on them briefly. This is to just say you should go back and read these papers, essentially, but it's slightly more than just listing their names at the bottom. So they take a network and train it to do a task that is also done in the non-human primate. And by reverse engineering or analyzing the dynamics produced by this trained network, they find that there are 2 different integrators, very similar to the task I'm talking about here, one for integrating motion and one for integrating color in this non-human primate task. The second example is done by Devika Narayan and Wang, whose first name I unfortunately can't remember, in Mehrdad Jazayeri's lab. And they trained networks that can, in a context-dependent manner, flexibly produce responses of 2 different speeds. And they've also compared that with biophysical data. I was kind of hoping that Devika would be able to join us today and give us a little primer on her paper. And that would have been really great because she's an extraordinary speaker, and it's something that you should keep an eye on. But anyway, here's a second example. The third example is from Abby Russo, who did this work in collaboration with Mark Churchland and Larry Abbott, and they trained a neural network model to reproduce activity from a macaque that was doing a cyclic task, so literally pedaling a monocycle kind of wheel. And they, I think, got at a fundamental problem in neuroscience, which is if you record activity from a region, how do you know that activity originated in that region or it came from somewhere else connected upstream to it? And to get at that question, they developed a new metric that they called untangling that tackled this problem. So this is like a little just saying, okay, here are 3 papers you should look at. And this is what I mean by mechanism, right? Can you get at a quantity or something that go beyond the details of that specific experiment or measurement? And so that's what all 3 do. They give you the answer to how some task was performed by the dynamics within a network. They tell you about something that you couldn't have got from measurements alone. And finally, all 3 papers use this concept that we've been talking about, which is the geometry of these dynamical motifs and simplified ways of visualizing and analyzing them in state spaces. Right, so where are we, right? We're trying to look at, we're enriching our theory of cognition as dynamics. I just told you that using a variety of learning rules, of which we saw 2 specific examples, we can go from something that was a randomly connected network to something that is much more structured, specialized to a task. So in the previous slide, you saw these 3, right? Those 3 J matrices have very little to do with one another, right? They're specialized. So you can't say very many general things about them. But the same sorts of analyses we did for visualizing, eigenvalue spectra of the J matrix in the random case can still be used to provide some intuition here. Like if David Susilo found integrators, then are there real eigenvalues in the stability matrix that correspond with no imaginary parts that correspond to those fixed points that he claims to have found? Answer, yes. And we wouldn't have known to do that if we didn't know that fixed points were a product of these types of real eigenvalues. So what we're on the route to doing is to train nonlinear RNNs like this to do something brain-like, to do so flexibly, to do the task in a rich dynamical regime to allow for these motifs to even appear in state spaces and still get repeatable, reliable performance from these networks. And I want to show you in much more detail in what's coming up, what else I mean by mechanism, right? So in depth, really looking at what types of mechanisms we can gain from these networks. And so what we're doing is taking our original theory of cognition as dynamics and enriching it with the concept of the geometry of these dynamical motifs in state space. So I want to take a beat here, see if there's any chat questions that I should look at or pay attention to before I get into this part of the talk. Nope, nothing. We're good, keep going. Somebody nod.
May I ask a brief question?
Please.
I was wondering, especially in the target-based method that you showed, like, is that related to generalized linear models? Is that an example of that?
They're not actually. So you can think of the error function as the way you write error functions can be boiled down to a least squares problem. So that's where the correspondence ends. But remember here, the linearity is broken by the P matrix, which is the inverse of the correlation of the full nonlinear dynamics. Right? If it was just the error function with some kind of regularizer, then it would look a lot like GLM. But this is neither general nor linear.
Okay.
Cool.
Thank you.
All right, so we're in this era of unprecedented technological advances in neuroscience, right? So when I was first coming up, we went from recording 1 neuron at a time to recording from populations at a time. So that's what I refer to as the first revolution in neuroscience. Now we're in this era where we're recording more and more neurons from many parts of the brain simultaneously during behaviors. And yes, we can get into a discussion afterwards about the wisdom of such an endeavor is more necessarily better. And I have controversial thoughts on the subject, but here we are nonetheless. Unprecedented neurotechnological advances are allowing us to record activity from many parts of the brain at once. So activity usually comes out either in spike trains or calcium imaging data, wide field imaging data, even voltage sensitive dyes from all over the brain. The data comes out in this high dimensional, kind of hard to parse way. I mean, we could barely parse what it looked like even in the population dynamics from this network that we built, right? So we had to cook up the state space idea, and we had to cook up looking at autocorrelations. So one way to look at something like this, since this lecture is about dynamical motifs, is to do state space analysis. So we could take activity that came out of experiments like that and do some kind of dimensionality reduction technique, derive ourselves a new coordinate frame, and repeatedly, many different papers, and I've highlighted only a few here, and there's just more and more, including some very good reviews. In fact, one by Shreya Saxena with kind of an unfortunate name, but really good review on these types of population methods. They called it population doctrine or something, which is what I was referring to as an unfortunate name. But there's a review of these methods by Shreya Saxena about looking at population-wide dynamics in this form. A lot of these papers have the following statement, which is that activity that is relevant to a certain task could occupy the full n dimensions available to it, but it doesn't. Even in our chaotic attractor, right? If you have a network of 1,000 neurons, the chaotic attractor, if you did state space view of that chaotic attractor, would live in about 30 dimensions or so. So it's not as low dimensional as the Lorentz attractor. It's still high dimensions, but it's not the full dimensionality available to it. So any task relevant computation over many different empirical systems seems to occupy these low dimensional manifolds in state space. The manifold can be described by looking at the various axes spanned by the covariance between these neurons. Covariance is nothing but basically, the covariance of R times RIRJ, basically, which is the activity of everyone, the cross correlation. The cross correlation of the activations is still a product of the outputs that people are recording, right? Your experimental colleagues have provided you activity patterns that they've recorded from these networks, and you've constructed the covariance, the axis of which gives you this neural manifold, and on we go. The question that still remains unanswered is, what are the inputs that are driving this covariance? So we looked at in the basic lecture and in today's lecture that these inputs can be external inputs from the outside, sensory stimuli, task-related feedback, and so on and so forth. But really, the inputs can also be interactions amongst brain areas in this, in the actual brain. So here's a little cartoon. We're, belying our physical sciences education by saying, let's pretend the neuron is an ideal sphere. But I'm showing you 3 possible areas out of many in the schematic. Area A, B, and C probably connected reciprocally to one another. And so it's brain-wide interactions that form the inputs that shape neural manifolds. But current techniques don't give us access to these inputs. So there are a few quantities or the need of the hour are inputs to each neuron from within and across brain areas. So if my neuron I was in brain area A, how much of my experience as neuron I is shaped by other interactions from region A versus recurrent interactions from somewhere else indirectly connected to area A, like area B or area C? What we don't have access to are also directionality of these interactions and the effect of common inputs. So first of all, a lot of covariance matrices are symmetric. So interactions cannot be assigned a directionality to. 2, I wouldn't know if area A is providing common input unidirectionally to area B or C, or are area B and C providing inputs convergently to area A. That is also inaccessible by just looking at the covariance. Finally, a lot of the subspace-based methods are not scalable to more than 2 regions at once. So they look at pairwise interactions only. So how do we address all of this in one fell swoop using the same building blocks that we saw before? So things aren't going to just ratchet up in complexity, by the way. That's all I want to reassure our young colleagues here for, is that we are using the exact same differential equation. Things aren't going to just spin out in the next slide. So what we can do is to build multi-region RNNs. to get at mechanisms that are inaccessible for measurements. What do I mean by mechanisms? Are exactly those needs. Can we look at effect of common inputs? Can we look at inter-area interactions? Can we look at directionality of these inputs? Can we really look at interactions beyond pairs of regions at once to potentially brain-wide networks? And so we can build multi-region models and we can train them to match data directly. So that's the approach that we're going to be taking today. So that's what I'm calling here constrained directly by neural data. You can constrain them by neural data, by behavioral data, or both. And in fact, there's now beautiful theoretical work that shows that the more constraints you have in terms of neural and behavior, the less you have to hope and pray you'll find a line attractor in state space. That there's less hope and prayer, and that if there is a solution to be found and multiple learning rules can get you there, then maybe biology found something analogous. It's a principle that Dan Yammons called contravariance. I haven't studied philosophy of mind, so I can't speak very much to it besides the word, but that's whose reference you want to go look at. But here, what we have done in my lab and in other labs that take this approach is to constrain network models by using other models, such as in teacher forcing or imitation learning. And that's what the gears on the top are supposed to symbolize. You can build these models that are constrained by data from small nervous systems where you have access to a large amount of data, such as the larval zebrafish, or you can build network models that are constrained by larger and more complex brains with much richer behavior repertoire, with the concomitant problem that the sampling is really crappy in those brains. So you can do all of these exercises. And yes, if I were a dyed-in-the-wool machine learning engineer types, then I would build the literal sampling ratios at the cost of burning down several rainforests to try and test if these things are worth doing or not. But I'm not. So what I will do instead is to tell you that if you have a few hundred units from about 2-3 regions all the way up to 13 regions in the larval zebrafish case, we can still make progress. We can still get at quantities that you couldn't get from measurements alone. And the route to getting those quantities runs through the ability to analyze these data-constrained RNNs using techniques that are very, very similar to stuff that experimentalists can do. So looking at state spaces, looking at angles between trajectories and state spaces, looking at dimensionality, looking at connectivity. And we're going to look at all of those things as we proceed today. Here are a few references. I will send you a reading list. Right. So this is where I can take another small beat before I get into the details of the larval zebrafish. So do you have anything pressing that can't wait? Actually, okay, so I stare blankly because I can't see I can only see like 5 faces on the side. So somebody's got to either wave frantically or say something for me to even see what's happening. Keep going.
Yeah, you guys should give some feedback.
Yeah, exactly. I'm like, have I lost everybody? I know it's a long thing, but cool. All right, okay. So these are experiments that were done by Aaron Andelman, a very talented postdoc in Carl Deiseroth's lab, who now, by the way, runs this extremely profitable startup. So there you have it. So the experiment he did involved taking larval zebrafish, head fixing them, and exposing them to this behavioral challenge experiment. So what he did was head fix them. Larval zebrafish don't do a whole lot except whip their tails. And at some point in the experiment, he starts applying electric shocks. Very small shocks. They appear not to really actually destroy the animal, believe it or not. So it's about 1.3 milliamps. And they're applied to the animal in this open loop fashion. When the shocks first appear, the animal whips its tail vigorously to try and evade it because it is still unpleasant. Now, this experiment goes on for a long time, about 20-25 minutes later. Since the animal perceives this as persistent and inescapable stress, it stops struggling. It goes into the state called passive coping, occasionally also known as learned helplessness. And this state has been thought to be an analogy in other nervous systems to be a marker for major depression. And the reason I like this is also because, I work at Mount Sinai. The next door neighbor of mine is Helen Neighbor, who pioneered deep brain stimulation for major depressive disease. And when I talked to her about this paradigm and these results, originally it's like, oh, yawn, kill me now. I want to talk about patients. But then she tells me about how these patients that appear for clinical trials have often movement problems. So mood disorders and movement disorders were considered dichotomous, but not anymore. So a lot of mood disorders manifest themselves in an inability to move. This is why learned helplessness as a field came up, because of its potential therapeutic whatever. So to my knowledge, this is the first time anyone had shown that such a thing can happen in larval zebrafish. Wildly ethologically irrelevant stimulus. So zebrafish live in these murky things, paddy fields in India, Pakistan, and Bangladesh, and like that. So the most stresses that they actually encounter in real life include sunlight and heat and salinity, pH, that kind of thing. Shocking is wildly unrealistic, but it provides us this. There's two behavioral states. There's an active coping state characterized by vigorous evasive response, followed by, after a long period of time, passive coping characterized by a full cessation of movement. And that's quantified over here. This is the speed of the tail whips as a function of time. In the pink bar that you see is the duration of the behavioral challenge. I would like to draw your attention to the time scales we're talking about here. So those of you who work with macaque data and mouse data will be surprised by this, because we're now talking about 30, 45-minute long experiments. And in black is average of the tail whips for control fish. In blue is that for the shock fish. And you see these two phases quite distinctly, right? The shocks come on, there's an elevation in the tail whips, which is where the blue is going over the black. And after a while, the blue sinks below the black, sometimes going all the way to zero. And that's the passive coping state. Now, because the Dazeroth lab is one of those labs where there's explosive neurotechnological advances, they're also able to image for us activity from the whole brain of the zebrafish during this entire experiential state. So in this line of zebrafish, they've expressed GCAMP in the nuclei. So they know that whatever is glowing is really an actual neuron, and they provide us activity from anywhere from 10 to 50,000 neurons per fish. For what I'm showing you here is on the left is data from 5 control and 5 shocked fish. So many fish over this long period of time. So these are sort of heroic experiments to have pulled off. But how do you go beyond look at our experiment? How do you design something that is a mechanism? How do you go from activity like this to explaining this behavioral transition? So I will tell you a couple of things. I will also tell you a failure mode of theorists like mine as we go along. So stay tuned. So why larval zebrafish? Because we want to be able to extract mechanisms and principles from smaller brains with more access. So it's got only 100,000 neurons total. You can see right through the animal. So it's this high throughput way. There's a lot of transgenic lines and optogenetics magic you can do with this animal. So you want to be able to extract some mechanisms from them. They nonetheless have many nuclei or brain areas from which activity can be recorded. Some of those nuclei have homologies with the mammalian system. So this active coping and passive coping have been studied extensively in mice and rats. And I'm showing you the cartoon of the mouse neurobiology here. And there's some regions that are analogous to regions that are found across both species. The behavior also has parallels. But the larval zebrafish system provides us even more access than the mouse system does. So what we want to be able to do is, together with the Dazeroth lab, ask, are there any mechanisms that are conserved when you go from one nervous system to the other? And as a theorist, I have the privilege to be able to ask this pretentious question. Let's say we have data in this active to passive coping paradigm from all of these nervous systems. Will I be able to write down a physics style model and say, here is the minimum multi-circuit model that mediates something like learned helplessness? Or will I just find that every species is its own unique snowflake? So identifying convergences and divergences are both sort of interesting. But from the perspective of today's experiment, let's restrict ourselves only to the larval zebrafish and figure out what in the neural activity patterns that you see brain-wide caused this behavior transition that you see from active coping to passive coping over this very long extended behavioral experiment. So here are a couple of things that they didn't need a theorist for, right? There's a couple of findings that just popped out of the measurement. And these popped out because they were looking for them in some sense. So in the homology picture that I showed you before, you had to pay attention to two nuclei. One is called habenula and one is called raphe. And we will return to those names over and over. So there were two experimental findings that just popped out of just the measurements. The first was that during the process of this behavioral challenge, activity in the habenula seemed to ramp up. So what I'm showing you here is average activity, average delta F over F for two pieces of the habenula, the ventral and the dorsal habenula. In black is the control fish, averaged over 5 individuals. Blue are shocked fish, averaged over 5 individuals. And you can see that they both have this sort of ramp-like activity. In dashed line is the time at which the shock is initially applied. So, and I'm going to tell you the story that we were about to build. Along with this activation in the habenula is a concomitant suppression of the activity in the raphe. Habenula and raphe are monosynaptically connected to one another. Habenula projects to the raphe does not project back. This is a known anatomical circuit in mice as well as in larval zebrafish. So this was kind of a nice thing. And let me tell you the theory that we were about to build. So, I swoop in and I go, we can write down a simple theory, no problem. So the theory we were going to write was something that smelled a lot like control theory. We said bad stuff happens. Habenula is the one that's going to say bad stuff's happening, bad stuff's happening, bad stuff's happening, punctuated, right? It's integrating the stress to a bound. At some point, the projection to the raphe gets activated. Raphe's job is to dump serotonin into the system and say, I hope you feel better. If you didn't feel better, the animal has to lapse into passive coping or it'll go into shock, right? So who shuts down the movement? We know from work in mammals that there's a thing in the prefrontal cortex called the infralimbic cortex that shuts down movement. So the story we were going to build is habenula says bad stuff's happening, complains to the raphe that says cavalry's here, don't worry. And that together sends a projection to the infralimbic cortex-like thing that shuts down the movement. So whatever it is that is shutting down the movement has to be correlated most tightly with the time at which the animal lapses into passive coping. So what we thought was going to happen, we've made this prediction. We know how this activity pattern should change in the brain. We're going to discover that for the first time, there's an infralimbic cortex in larval zebrafish, fame and fortune. Answer, that is not what happened. So the theory that we had built turned out to be wrong. So let me tell you what we discovered anyway, even though this elegant theory that I wrote down just kind of crashed and burned. So to get at this, what we wanted to do was, the pursuit to finding this infralimbic cortex. So I'm going to tell you how we proceeded, right? The first thing we did was to go from a single module RNN to multi-region RNNs. And this is the same schematic that you've seen 1,000 times before, model units connected with recurrent connections. And we turned them into multi-region RNNs by wiring multiple ones of them together. We're now writing a paper with 13 of these connected reciprocally to one another. But here I'm only showing you 3, RNN A, B, and C, connected through inter-area projections. Now, the only difference between the first thing and the second thing is that in the first case, the connectivity matrix or the recurrent interactions come out in this, one distribution where the columns indicate the sources or the presynaptic guys, rows indicate the postsynaptic or target guys. But in the multi-region RNN, you can further parcellate this matrix into its components. So what I mean by that is, if I've constructed a network in which the inter-area projections are, let's say, sparser, as is indicated by the schematic here, then along the principal diagonal, you should be able to identify recurrent connections within RNN A in blue, within RNN B in yellow, and within RNN C in red. And if you wanted to look at projections from B to A, you would just look at the block right next to it. If you wanted to look at RNN C to A interactions, you would look at that submatrix. So it gives you this compact mathematical representation that still has all the advantages of working with matrices, but it also lets you take out submatrices and analyze them separately. And this is going to become super important to us as we go along. So I'm not just telling you this for the fun of it. The second thing we do is to train this network to do something. So when you start this network off, it's just going to produce chaotic activity because it is still a large network that's connected reciprocally to one another, initialized randomly, right? So what we want to do here, because we have data from everybody, we can assign a target function to every neuron in a network like this, and we can write down a learning error, which is exactly the same thing as I showed you before, which is Zi minus Fi, which is the linear difference, instantaneous linear difference between the neuron where it is and where it needs to be based on the target from data. And at every time step, the entire brain-wide matrix is undergoing training so as to autonomously produce activity. That's the goal here. So the first thing we did was to go from single module to multiple regions. The second thing we do is to take the activity of everybody and train them to match data directly. Now, for the purposes of today's talk, there's a one-to-one correspondence between the number of units and the number of targets. But in our papers, you will find that we've also built much smaller networks and embedded them into larger networks, or we can build much larger networks and subsample as few as 10% of them and still get comparable performance because these are very, very powerful networks. So what does this activity buy you, right? Have we really built something that is as complex as the larval zebrafish and equally uninterpretable? And I'm here to explain to you that that's not true. So what are the various outputs you will get from a network like this, right? The various outputs from these data-constrained multi-region RNNs are, once the training is finished, you have a network model that produces realistic neural dynamics autonomously. Because if you know the J and you know the initial condition, X dot is minus X plus J phi should let you generate this activity in perpetuity. And that's indeed what happens. That by itself is not surprising, although, well, I don't know. Let me just tell you what the advantages are, right? It's not surprising that it does so because that's what we're training the network to do. It is surprising that it has significant advantages over functional connectivity, which is the stability of the dynamical system. So what do I mean by that, right? All N neurons are being trained at every time point. If you ignore the biological possibility of this training rule, you're getting both the on manifold directions and all the off manifold directions right. If you were only fitting the covariance of the activity, you will only get the on manifold right, which means that you have no guarantee of stability. You're getting all the off manifold directions right. For all low dimensional dynamics, the number of off manifold directions are vastly more, which means that if you perturb the system from on manifold to off manifolds, it should still attract back. And that's what this kind of system gives you. So yes, it is not surprising that it gives you the dynamics, but it is surprising that you get stability for free. The second thing that you get from these data-constrained RNNs is the ability to infer from them these brain-wide interactions by looking at the trained J matrix and its subblocks. That is not something you could have gotten from experiments alone, although one might make a very tenuous argument to data that is coming out now from various EM-based connectomics. At least it will make some kind of prediction for those types of studies. But right now, I'm not able to say much more than hand wave through them. But you're able to infer these matrices consistently. So what do I mean by consistency here? I don't mean that if I tell you neuron 455 and 500 are connected by 1,000-fold strong synapses and you go in and you find them. So point by point, those entries in the matrix are not going to line up. But you'll still get the statistical properties of groups of synapses right, which means that even if I start from a wildly different initial condition or use an entirely different learning rule, I will still get a distribution that looks the same or similar. And I will show you an actual example of such a thing. So this consistency is a powerful feature. It is not exact though, and that causes a small problem, which led us to discover the third thing. The third thing to me is actually the most surprising of the first two, which is the product of those two objects is J phi. You have a trained J matrix and you have these activations, right? The product of those two objects gives you currents due to recurrence. Just by summing over this little J index, you can extract currents that region A is experiencing from other neurons in region A versus currents that it's experiencing from B and currents that it's experiencing from C. So let's look at these pieces a little bit more, right? So here's, I'm going to show you a 3-region RNN, because just slapping 13 regions on you in a summer school is just rude. So here's 3 regions. Remember that in the story, we were trying to find something that looked like an infralimbic cortex. So one of the regions is going to be modeled after the habenula. One of the regions is going to be modeled after the raphe. And the third region is going to be the telencephalon. Because, it's a cortex-like blob, it had the most units, and so it checked off 2 of my requirements, right? I was looking for something that was like a cortex, and I'm also trying to figure out the effect of common inputs. So are A and B mutually interacting with C, or is B the common input to area A and C? Questions of that nature. Telencephalon seemed like a convenient choice. So this is where they appear in the brain of the fish. Telencephalon and habenula are not subsets, they're just in different Z planes. And here's a little cartoon that tells you where stuff is. Now, networks like this work extraordinarily well. They kind of almost always converge if you have continuous data like this. So here's neural activity, and I apologize, these right-hand side panels should really appear, but I took them off last night to answer some discussion questions and I forgot to put them back on. So here's activity from 3 model units. A habenula-like unit in blue, raphe-like unit in red, and a telencephalon-like unit in yellow. I'm showing you the delta F over F of 1 example each. In black is the actual data. In blue, red, and yellow are the network's fits. And you can see that the network produces a slightly denoised version. It gets the overall dynamics right, but it does miss the high frequency bits. And so we're sort of getting a washout effect. And that's just the nature of networks like this. So they're going to miss, if these little correlations between the temporally sparse peaks in the habenula and raphe were important, then we're going to miss them completely. But we get the overall flavor of the dynamics, right? And I chose to keep that flavor because we're looking at an experiment that's like a half hour, 45 minutes long anyway. So I was like, it's okay if we're missing these. Now, just to convince you that I didn't cherry pick 3 examples here, I'm showing you the average, the activity projected into the dominant principal components on the top right. In black is the data, and in red is the model. And you can see here too, right? Like the data kind of has these like little idiosyncrasies and these jagged bits from the biology, but the network gets the overall population dynamics right. And at the bottom right, you should see the variance explained as a function of the number of principal components. And the model gets quite a few of them right, but we're not, at the tail end. So we're still getting about 15 or 20 principal components of the activity. So once the network is trained, it's going to keep producing this activity forever. This by itself, not that surprising, the stability of it surprising. But really what models like this buy you are the ability to look under the hood, right? This is not stuff that you're able to do from measurements alone, not in, at least in the larval zebrafish case. So what we did was to look at the overall histogram of everybody in this 3-region network. And I've taken a log scale on the y-axis because the range was weird. And I've normalized everybody so they all start at 1. So your eye has something to look at. It's a purely visualization trick for you. Everybody ended up having a mean of 0. And in gray is the J matrix of this 3-region network corresponding to a control fish. And in blue, that corresponding to a shocked fish. Now, if you all remember your first basic lecture, the randomly initialized J is normalized to G squared over N. So let's say for arguments and purposes 1 over N. That means that the Gaussian initialized matrix is this very small U-shaped structure, about a 1/4 of the way into the middle. So already, even in the control condition, there's a huge expansion in scale. So what you see as a Gaussian initialized becomes this exponentially distributed distribution. Additionally, there's an even further expansion in the strength of these connections in the shocked fish relative to the control fish. The third feature here is that rather than being governed at random, tuning in the control fish and the shocked fish appeared to be by means of very large, very few synapses, because they have very low probability and they're way on the extremes of the x-axis. So these are useful features for us to know. The means all came out 0 rather than some mechanism pushing everybody to the right or everybody to the left. The kurtosis and the skewness, which are the 3rd and the 4th moment, did not show appreciable differences. I'm showing you here just the average over 5 different individuals, but there's a small spread around each of them because there's some individual to individual variability. Now, this matrix also lets us look at submatrices, right? Because that's the advantage. So where is this expansion in synaptic strengths coming from? So we now have the ability to take this network and really look at the distribution of weights within the subblocks. Now, we were interested really in the habenula and the raphe first. So those are the results I'm going to show you here. And we're going to go into much more detail in a second. So we can look at connectivity within the habenula block, which I've illustrated here in the schematic. And indeed,
Kanaka, I'm so sorry to interject. Whenever, if you want, we could take a break whenever you think is a good moment. There was no obvious good moment to
maybe after this I could
ask you, but whenever you want, whenever you want.
But aren't the students the ones that are more exhausted than I am? Well, wait, let me see where I am, right? Yeah, you know, let me finish this and then it's time to take a break.
Sure, whenever is convenient.
Okay, I mean, I could keep going and then have a longer discussion, whatever you want.
It's as
depends
you
on the exhaustion level here. You guys have to weigh in if you want your opinion heard, you know.
Yes, I think it's as you guys want, as you prefer, Kanaka. Anyway, why don't you finish this section and then you can ask and we can see what's best.
Will do. Let me do the slide and then I will ask again. But really, if you don't vote, you don't get to complain after. So really the bulk of the changes in the connectivity matrix were indeed coming from the habenula. Not super surprising, very thrilling to me because remember the story, right? Oh, habenula is making this amazing ramp. Maybe the ramp is caused by these like strong connections within the habenula. So the habenular block indeed shows this expansion. The blue is bigger than the gray one. Now I wanted to look at the other blocks, right? The habenula to raphe projections because I'm still constructing this theory in my head. And that did not move me to tears. Within raphe connections, if I kind of decouple my eyeballs and squint hard, I can pretend that the blue is slightly smaller than the gray. And so it may probably underlie the inhibition that you saw in the raphe, but again, did not move me to tears. Here's the really weird thing that destroyed the theory though. The connections from the raphe to the habenula appeared to be the second biggest driver. So they also showed this strengthening of these projections. Now, raphe to habenula, there is no known anatomical monosynaptic connections going from the raphe to the habenula at all. That said, we know that these networks have very slow dynamics. We're washing out a lot of the fast effects, and raphe's job is to dump serotonin kind of brain-wide. So maybe we're picking up some kind of indirect effect here, but this is an example of something that the Deiseroth lab is now testing experimentally. So we're basically saying there was a ramp in the habenula that is caused by strong connections in the habenula. So optogenetically deactivate the habenula and see if this goes away. 2, second experiment that they weren't originally planning to do, but they will do now because of a broken theory is to block any potential feedback from the raphe to the habenula or localize the serotonin release in the raphe and see if this effect goes away. Or maybe we have discovered some kind of connection from the raphe to the habenula. Now, there's one paper in the mouse literature by this person called Schobel et al. That shows that there may be a GABAergic projection going from the raphe back into the habenula, but it's tenuous because there's literally only one paper. But anyway, this is the kind of unexpected finding that could come out of building a network model like this. So at this point, I want to take, I want to ask you guys last call, because after this, we have to blast through to the end or it's not going to make sense. So we can take 5 minutes now, or I can keep going.
So you guys should say something.
5 minutes.
Okay. Okay, first come, first serve, I guess. So let's take 5 minutes. We'll see you at 20 past. How's that?
Sounds good.
Please, guys, interrupt whenever you have a question. Don't hesitate to interrupt. I know we're all a bit tired, but you should speak up. Please, go ahead.
Hi, Kanaka. Can I have a
basic question.
Sure, ask me.
One is what's the difference between the multiple population model and multiple ensemble model with here the multiple region model RN?
I'm sorry, I didn't follow. Can you say that one more time, please?
I cannot clearly distinguish with the multiple population model RN and multiple ensemble RN with your multiple region RN.
So I don't know what those first two things are.
I'm not sure whether all the neurons within the same region, they share some identical or share some similar statistical properties. So
I see what you're asking.
Can they be divided into the same population or the same ensembles?
Got it. Okay, so you're asking kind of actually, yeah, very good question. Okay, so we're going in this paper, we went with the area definitions that were given to us by our experimental colleagues. So they had already used clustering methods to figure out what was an area along with anatomical markers in the larval zebrafish. So they had already told us that neurons with this particular index, I know 2,200 neurons from this particular index were all habenular neurons. So that's the kind of assumption that we went in with. We didn't independently cluster the data based on what we thought was statistically similar. Although in the next section, you will see that you can take the same, what we think is one uniform area, and you can start to look at things like submodules within the same area based on statistical properties. But in the models that I'm showing you results from, everybody's connected all to all. So we haven't made any special considerations or any assumptions for anybody belonging to any region. All the neurons, in addition, receive this kind of pulsatile input with the pulses delivered at the same times at the times of the shocks in the actual experiment. That's literally the only correspondence we kept, because we wanted to see first order what happens.
Okay, thank you.
Cool. But yeah, the question of what makes an area is a good one, because I'm going to show you something that will then make us question such a thing. Right, so what I've told you now is, okay, we can get activity. So I told you that we get three possible outputs from these things, right? First, we can get activity that looks a lot like the activity that you were training to match it. You can also look under the hood and extract these types of connectivity profiles, which you can then use as probably priors to build simplified models, to look for things from connectomics literature as more and more connectomes like EM-based connectomes start to come online. The third piece I had already hinted to you before is this thing that we're calling current-based decomposition, or curbed for short. And that comes from the observation that networks like this give you both those objects, right? You get activations that are similar to data, or you get the set of phi's, one for each unit in this network, and you get this directed interaction matrix, which I'm calling JM here. And a matrix from a network like this contains connectivity within the habenula in this blue block, within the telencephalon in yellow block, within the raphe in this red block, but also experience of the habenula from neurons in the telencephalon and from the raphe. You can also get all the other interactions like this 9-way interaction map from looking at the other submodules. But for today's discussion, I'm going to restrict myself to just the habenular experience, because that's the one that I was the most keen to understand. We can also analyze the rest if you want. But the idea here is that the dot product J phi, just by summing over this little index J over habenular neurons or telencephalon neurons or raphe neurons, is going to give you 3 currents. The sum of those currents still gives you the activation you recorded. But the J allows you to decompose this activity into its component source currents. So what do I mean by that, right? Like activity collected in the habenula can now be demixed based on this matrix into 3 currents. So this is the same exact ordering in these 3 plots that you see. 2,200 habenula neurons in this one particular shocked fish as a function of time, habenula to habenula currents, raphe to habenula currents, and telencephalon to habenula currents. I've just sorted the habenula to habenula currents because I was kind of sick of looking at pseudo color plots that look like static, but the ordering here is completely like just for a visual thing. It is meaningless. So essentially, this decomposition lets you have access to a within area current, right? By just looking at this dot product habenula to habenula, looking at two separate inter-area currents, raphe to habenula and telencephalon to habenula, just by looking at which indices you're summing over. The sum of these three still gives you the activity that you recorded or your experimental colleagues recorded. But it now decomposes this activity and gives you a much more informative picture than just looking at the average and saying, oh, here's a ramp, or just sorting activity and saying, here's a sequence. Now, what I'm about to show you is you can also look at dynamical motifs and understand a geometric perspective of these dynamical motifs, except now you can do this in the current space. So what I've done here is to do PCA on these three currents and taken the dominant principal component from each, and that automatically comes out orthogonal, but if it didn't, you can orthogonalize them. And that gives you a new coordinate axis now in the current space. Habenula to habenula in blue, raphe to habenula in red, and telencephalon to habenula in yellow. Then I can project the activity of the habenula into this space. So now I'm projecting the activity of this entire ensemble into this coordinate space, spanned by the dominant principal component of the currents. I've also taken the times at which shocks were applied, and I put them in dots. Warm colors are the early part of the experiment. Cold colors are the later part of the experiment. Remember the story we were building? Habenula is the thing that's supposed to come on first. Driven by strong connections in the habenula, that's the one that is supposed to sense the behavioral stress. In fact, that is not what we see here. What we see here is that the early part of the activity is dominated by activity in the raphe to habenula current space. And it's only later on that the habenula to habenula or telencephalon to habenula currents come on. So this already throws a wrench into our favorite theory, that rather than being habenular or intrinsic to the habenular current, initial activity or active coping seems to be much more dominated by rotations to the raphe to habenular space than to these other two subspaces. So we can take this now and correlate it with active coping and passive coping now from behavioral data. And I'm now trying to convince you that this way of looking at activity by decomposing it into its source currents is much more informative, or at least can be a complement to more traditional ways of looking at outputs. For example, taking outputs and sorting them. I have written papers myself on sequences, or looking at activity and averaging them over individuals and making up a whole theory about trying to look for fixed points based on this ramp. Or in fact, even taking the activity and projecting it into the principal component space. Yes, you get some kind of squiggle in state space. Yes, you can write a paper based on a squiggle in state space, but the squiggle is much harder to interpret. The squiggle on the right, on the other hand, tells you exactly what's happening in which current. So it's at least a complement to these traditional points of view. Now, never trust a 3D plot, my advisor would always say, right? So I want to show you what these look like as a function of time. He also said weird things like never trust a log plot, for example. So anyway, here's these currents as a function of time. Here's an example from a control fish. Every time the fish whips a tail, it breaks a light beam. And so you get a vector of zeros and ones corresponding to every time the light beam was broken. That's the movement index for something like this. Another advantage of working with a smaller nervous system is the impoverished behavior, but impoverished is just code for easy to quantify. I've taken that vectors of zeros and ones and I've convolved them with a Gaussian, again, to give your eye something to look at, because I was sick of putting dots on this plot, basically. So in black is actually the movement in the control fish. In red is the raphe to habenula current, blue is the habenula current, and yellow is the telencephalon to habenula current. And you see that in the control fish, which is not experiencing any shocks, every once in a while, it's kind of whipping its tail, and the three currents are doing what they're doing. The same plot now for an example shocked fish is drastically different. So again, in black is the movement. This fish is not even a particularly well-behaved shark fish, because after lapsing into passivity somewhere around the 30-second point, it still whips its tail once for the heck of it. So what you see here, however, is that active coping in which the movement is enhanced is driven by a ramp in the raphe to habenula current. And it's only much later, in fact, after the fish has ostensibly lapsed into passivity, that the habenula and telencephalon to habenula currents come on. And that was basically the time version plot of the 3D plot that I showed you. Now and this effect is also conserved when you look at data that is averaged over multiple individuals. This should impress you because, as we know, there's a large animal-to-animal variability. There's a second source of variability in my initialization of the J matrices, right? And so all of those effects are still preserved despite those two sources of variability. So what you see here, once again, is that on average in shark fish, the active coping seems to be mediated by rotations in the current space external to the habenula, specifically from the raphe to habenula. And it's only later that other currents appear to come on. Now we're repeating this curved analysis with all 13 source currents into the habenula, and this effect is still preserved, followed by a few other kind of weirdly unexpected regions that seem to pop out and show different effects at different times. Now, to really convince you that there's two things happening, I have to convince you one other thing is not happening. And that's what this graph shows you. So now I'm going back to look at the J matrices derived for successive epochs in the experiment. And now I'm only going to look at the raphe to habenular subblock, because that's the one I'm kind of the most interested in. And I'm showing you the log probability density as a function of interaction strength. And I'm showing you three histograms here. In black is a histogram derived for this submatrix in the baseline period. The red is the initial part of the shock, and in blue is the later part of the shock or after passive coping. So between baseline and the initial part of the shock, the histogram did not move significantly. So in fact, this current effect of the early ramp of the raphe to habenula current seems to come only from the currents and not driven by any kind of mechanism that changed the J, at least in this subblock. It's only after the lapse into passivity that this change seemed to be dialed in that the blue histogram has much stronger connections than either the red or the black. So what we're saying is, so in the first paper, right, we were writing this other theory, which is to say, okay, habenular interactions ramp, and that ramp is caused by feedback interactions from the raphe to habenula strengthening, as well as strong connections within the habenula. Then we found that, okay, if we start to look at different types of time scales in dynamics, in the current space, and in the behavior space, we start to uncover different roles of projections from other areas into the habenula. And that's what we're writing up now. We're saying that there may be multiple time scales, and those time scales can be deplexed if we look at these types of patterns in the current space rather than looking at it in the traditional state space view. But once again, what I want to emphasize is this notion of the geometry of these dynamical motifs. And remember, even in the simple equation, you have activations as dynamical motifs. You have nonlinear dynamics in the current space, which you can look at as dynamical motifs. But if you went back to this matrix here, you can derive these matrices for chunks of time in the long experiment. So you can now derive geometric motifs in state space in the connectivity itself. So there's multiple scales at which you can look at the same sorts of projections in state space. So, right, blah, blah. This is just sort of, telling you a few more advantages of this kind of thing, totally PR. But here we're back to the lesson plan. And before I say thank you, I would like to tell you very briefly that what I hope you got away from the first lecture was an understanding of some of the foundational elements of RNN models, that I hope you left with an appreciation of their mathematical tractability and some of the impressive stuff you can learn about the general properties of them, even if they don't connect to biology directly. In the advanced lecture, I hope that I convinced you that you're able to make a tighter connection to biology by literally using biology, and that you're able to understand something about the biological brain by reverse engineering these toy models that are built to fit time-varying dynamics collected empirically. So with that, I would like to open the floor for discussion, and thank you all for your attention. Thank you.
Well, thank you very, very much, Kanaka. I think it was very, very appropriate lecture to end the school, because it's kind of opening a door on many new developments. So that was very nice. So any questions?
Last night I was thinking, okay, do I hate them enough to do this whole like mean field thing for them? And I was like, no, it's the last day of the school. Let's be nice.
I would have liked seeing it, actually.
Would you though? But you would be the only one. They would have all lapsed into a coma.
I don't know, but no, but this was very nice. So we will have to invite you to give that mean field theory lecture some other time.
Hard pass, not on Zoom.
Sure. Actually, where is it written?
What is?
This the analytical treatment. This mean field theory.
I mean, my notebook.
Oh, I see. So it's not something you've published.
Oh, you mean the lecture? I mean, I was going to give a lecture on mean field methods. I mean, it's a summer school.
Oh, mean field methods in general, not
in general. Yeah.
No,
not particular to these. No.
I see.
Because these are not in the mean field limit at all.
Yeah, right.
I mean,
right.
Exactly. They're not in the mean field limit. The matrices don't look, in fact, for the fish's case, if you look at the eigenvalues, they kind of look like they're making some kind of orthonormal basis set. They don't even look like they're random. There is a bulk that's random still, but but yeah, please bring up whatever you want to bring up.
Questions?
I have somewhat general question, maybe. I don't know if I missed that. Do you work with experimentalists on basal ganglia data? Because it looks like it could be an interesting fit for that, because it's a lot of interconnected subnuclei, which are part of a big subcortical loop. And it's kind of difficult to find those like mechanisms just based on the data that we have right now. And it's interesting.
I don't actually. In fact, this is why, I mean, somebody should have asked me, why are the mice red in all my slides? The mice are red because we don't have anyone working in mice data really. I mean, I have a collaborator in Chris Harvey's lab, but it's a very different kind of question because they are tackling more like wide field imaging. So they are much more interested in looking at sort of cortex wide things. But I don't have a collaborator in terms of the basal ganglia at all. In fact, Carl Swoboda, when I talked at Janelia, was talking, he's the one that planted this term in my head, interdigitated submodules within an area. So he's like, you know, weird areas like OFC, ALM, probably basal ganglia also. We think of them as these uniform blobs, but really functionally on the basis of these currents, you might be able to cluster them into different currents. So no, I don't, but I would love to follow up with you if you have that kind of bandwidth.
Like the data that we're working with, I don't think it would be suitable because it's neurophysiology, like it's electrophysiology. It's
I mean, it doesn't matter, right? These don't matter. It can be whatever, right? Like we work with ePhys data from humans and macaques. The same exact.
How many neurons would you have per like?
So a few hundreds is ideal. Really, right? Like 100, 200, that order. Anything sparser than that, and then I'm not able to tell you what you would need to know. So it has to be, so when people talk about sparseness, they're usually talking about the number of units, but it's also how much of it you have. So it's not an n by n problem so much as an n by time problem also. So like when I work with human data, for example, people give me ECOG arrays and there's very few of them typically. But those ECOG arrays go on for many, many, many days worth of data or many hours worth of data. So then that's still, the network considers them still sample data points, right? So that suffices. So if you have few neurons, then a longer recording is better. But if you have both few neurons and very short recordings, then these networks won't do much. Well, they will do something, but you won't get anything meaningful from them.
Yes, I see. It's also, it's relevant to what you mentioned, that you talked with a colleague of yours that depression and movement issues are correlated. Basal ganglia, like theorized to be involved in those impairments, like together, especially, for example, in Parkinson, what people talk about is movement issues, but it's also a lot of cognitive depressions, and basal ganglia is thought to be the kind of the centric part there.
Right.
Yeah.
Yeah, no, please, if you have any leads, then send it my way. I mean, there's other things, right, that are fascinating about the basal ganglia. Like, for example, if I were to go into such depth to look at modules within an area, I would put much more biological detail up front. Like, let's say I was doing that game with something like the striatum, I would make that an inhibition dominated circuit.
Which is basal ganglia.
Exactly, right? And so here, right, the advantage here is that there's two halves to the J matrix, right? So you can look at excitatory and inhibitory currents separately too, which we have in this manuscript that we've put on bioRxiv. I mean, at some point I stopped updating that bioRxiv paper, but there it is. It's got some excitatory inhibitory effects sort of separately. And there seems to be a asymmetry in some examples.
Could you somewhat moderate the excitatory inputs to be the modulatory dopamine inputs instead of just
I would like to very much, but haven't, because I don't have clever ideas for what to do with modulators, frankly. Right? Like for neuromodulators, my cleverest idea seems to be a very slow sine wave, and that's clearly not accurate. So, but yeah, I mean, we could make predictions, right? Like, to me, the coolest experiment is something that actually, I wish that I can claim credit for it. Somebody took the code base from the curve that's sitting on GitHub and they adapted it. So they were looking at a third region that connected to a pair of regions like V1 and LM. They said they were recording from already, and they were looking for a third region, but they didn't know where. So what they did was to take a 2-region network model that is fit exactly like I'm teaching them how to fit it. And the third blob is this unknown region that has random connectivity. So they first take this double network and they fit its activity to the activity they collect. They take the readouts from all 3 regions and fit it to minute-by-minute behavior or second-by-second behavior over a long period of time. And then they look at how the activity of this third region that looks like it's random initially is being modulated because of this training process. And the claim is that they know that there's a cognitive-like signal in it. Now you see how my words are getting more and more vague as I keep talking. It's because you're plumbing the depths of my ignorance here. But this idea to say, here's a signal I know should be there, but I don't know where to record from it. A method like this can tell you, here's the missing input, and that's the time scale it should vary at. That at least gives the experimentalist a place to look. Welcome.
Other questions?
Hey, thanks for the class and the class before. Today's class just made me remember something that I heard from psychology. You know, sometimes we can just start hearing this ticking of clock, and we cannot stop hearing it. And after a while, it just disappears.
Right.
And some psychologist friends of mine just told me the connection between them are just affected due to that, but he just didn't really explain it to me. Maybe you can apply your theory to that kind of experiments. I don't know, but
possibly.
That just came into mind.
Possibly. So human data are, I am learning, very tricky to work with for a variety of reasons. And some of those are just my limitations. Some of those is the fact that a lot of human data from control conditions are like fMRI data. And I just don't know what to make of them, frankly. So I need like somebody who's an expert there to sit there and say, okay, this is the work you need to look at. And, this is what it means when you're looking at voxel intensities over time and so on. So that's one sort of problem. The other problem is that ECOG array data is spatially very sparse. So there's only limited information that you can get from those patients. That said, I have a collaborator in Uli Rutishauser who's at Cedars-Sinai, another Sinai out at in California somewhere. So he has patients that have these brush-like electrodes. So there are these patients who show up with intractable epilepsy. And so because of this intractable epilepsy, they also develop depression, not at all surprisingly. So they have these movement motor disorders and like that. And they have these brush-like electrodes, so like a string and a brush at the end of it. So they're actually able to isolate single units from multiple regions while these people are just sitting there in the hospital waiting for a seizure. Because the goal is to identify where the seizure occurs and then to cut it out. So if you ignore the fact that these are patients that have invasive brain surgery, that there are baseline lots of drugs in their system, they can actually do tasks for you, right? Like they're so bored sitting in the hospital for days that they will do tasks for you. Like they will literally do tasks like, here are 2 images. Have you seen this image before? Is this a familiar image or a novel image? They will do N-back tasks for you, tasks that people do in rodents as well. So those types of data sets are the best suited because you get a lot of data from these patients over days. You can track the activity of these, networks that I infer over long periods of time. But, directly connecting to the type of behavior data that you're talking about is trickier.
Yeah, but sound can just be used as a stimuli for a mouse or graph, yeah.
So whose work you should look at is David Schneider. He's at NYU. He and I were in grad school together. So he works on the suppression of intrinsic sounds or something, right? Like sometimes if I'm like, I don't know, if you have too much coffee, you can kind of hear this like vein going off in the back of your neck or something. And then after a while, you stop paying attention to it. So is it habituation? Like you just can't hear it because your sensory sensitivity has gone down? Or is it some kind of active suppression of self-generated signal? He has a whole paper on the latter, that it's not habituation. Like in the, it says it's actually quite similar to this thing that's happening to the fish, that it's not habituation. The fish aren't just sick of the shocks. There's an active transform because they recover when you put it in a different context, for example. That's an important control. So
thank you.
Welcome.
Other questions or comments?
Questions, comments, criticisms, I no claim that this is a one-size-fits-all model. It's just something I like.
I was wondering if, you probably know this kind of literature, just these questions that people ask about whether something is learnable over time or has to be to some extent hard-coded genetically. And I was wondering if fitting your kind of models would help for these kinds of questions, if you know examples, if you have ideas related to that.
I have ideas related to it. They're not very good ideas, unfortunately. So I can tell you some. So I am, sort of the models that I trained to meet data, right, are kind of starting from this random wiring. So people like Tony Zader, for example, would not like them because he thinks that there's so much innate prior information already in the connectivity matrices that starting these networks or anything in ML basically from a random initial condition doesn't make sense. And initial conditions do make a difference to where you end up. So that's one answer. The other answer, however, is kind of, does it matter, right? So let's say I have, let's say this animal that's learning a song from its parent, like the songbird literature, right? This animal is kind of, it's born with a lot of structure in its head, sure, but then it's learning its parent's song through imitation. In that sense, what you have is many, many days worth of data, right? So what you can do in that case is to chunk the data into successive epochs and then take a network that is trained on a particular epoch and use that to seed the next stage, right? So rather than taking chunks of data and training everybody from random initial conditions, you can in some sense chain them together. So now I'm saying chain and train and they sound kind of the same, but I'm hoping you can hear the difference, right? So you can actually chain successive RNNs by making sure the initial condition is the one from the previous epoch. So then the effect and the more finely you're graining this epoch, and the more epochs you have, the less the initial randomization matters, right? Because you're looking over such a long period of time that kind of who cares? So my answer to your thing is no, we cannot capture the, the complexity of the evolution and like that, because the hardware and the software are both changing. But within tractable limits, the learning of a task can certainly be captured from this approach.
This is a related question. I wanted to know if you make several fittings on the same data, how much variability do you observe in the inferred connections? The
larger the network, the smaller the variability. Okay. And we have an answer to that a little bit already, right? Because these networks.
Is there some underlying similarity? Like, can you say something on that?
Yeah, so this came up yesterday. Louis Tao asked me this about, universality classes.
Yeah, exactly. Because there was this paper from Susilo and
So I am very hesitant to make any claims of universality when it comes to biology. Because I think that it's not a term that you can, it means something very systematic, right? It's not something that says, okay, I tried 2 different learning rules, I got the same thing, so whatever. It can't be a whatever. So universality is kind of a very tall order.
Okay, yeah.
In my case, even if you vary the learning rules or you change the initial conditions or subsample or hypersample, the larval zebrafish system always seems to converge to these exponential distributions.
Okay.
But exponential distributions are not universal because in the mouse case, they don't look exponential. In the human case, they don't look exponential either. So it's not a peculiarity of training these RNNs that somehow because of the nonlinearity or whatever, they're going to get there.
Okay. And how much influence do unobserved inputs would have in the results?
It depends on the system. So like this example that I was telling Kate before, right, about this unobserved area that is somewhere, they know it plays a role in the behavior. So then they fit a randomly connected network there and sequentially train it after they train all the other bits to see if that thing can rise to capture the remaining variability. But the best answer we've got for that is to take a network like I showed you and embed it in a much larger network. Yeah. And the result is interesting though. So the more complex the task is, the more helpful auxiliary units appear to be. So let me give you a more concrete example. So Chris Harvey in his lab trains mice to perform this task. So the mouse is in this T-maze-like thing. It says it sees a blue corridor, it goes left. It sees a red corridor, it goes right. This is rule A. Halfway through the task, the rules switch. Blue now means right, red now means left. Okay, rule A and rule B. This is a complex task for the mouse. And I'm saying complex, not in the algorithmic sense at all. I just mean difficult for the mouse. It takes them a long time to learn this task. Then there's an easy task that they occasionally give the mouse in the middle, where it's still the same T-maze. The wall can be colored whatever, but at the end, there's a big board that says go here. So it's super easy. The mouse doesn't even have to see the walls. It just has to head towards that signal. So there's an easy block of trials, and there's a hard block of trials, which involves these 2 rules that have switched. The animal has to grok that it has switched based on the reward thing, right? Like a performance will drop when the rule switches and then kind of comes back on. So when you fit networks to these data, so what we want to do is to see if fit the network on single trial data. There's many, many trials. We don't want a trial average. So we fit these networks on trial by trial by trial by trial. The easy task, the network can do fine. The hard task, an RNN with 1,000 units will struggle with if I train it with, let's say, backprop, because it's a time extended trial and there's many trials. So those gradients kind of vanish. So the gradients don't propagate if I don't use auxiliary units. So if I take 1,000 units, I train the connections within them to data, or I train them with backprop to the direct behavioral output, and I embed that entire network in 10,000 auxiliary units, which are not targeted, not trained, not plastic. Then the network can learn the task. So the more complex the task is, it's almost like it needs higher dimensions or something to decorrelate the input and do it. And I'm saying this vaguely because we're only now trying to understand like what's happening. Like, why are the auxiliary units helping in one case and not making a difference in the other case? So we're treating these auxiliary units as unobserved inputs or unobserved neurons that are nonetheless currently connected. And so we're playing with various combinations, like train the input weights, don't train the input weights, train the auxiliary units, don't train them. But in all of these
they are not plastic mainly.
They're not plastic.
Okay.
They're just adding this kind of specific variety of high dimensional noise through this J to the network. So just adding noisy inputs doesn't seem to help. So there's something about the shape of the attractor that is changed because of these auxiliary units that helps. So now, of course, the question is, just because the network's doing it doesn't mean the mouse has to be doing it. But that's, this is why they make experimentalists. That's his problem.
Just one quick, maybe I misunderstood, but you said there is less variability when the network is larger. That's a bit counterintuitive, because you would think for larger, so maybe you can explain the intuition.
So the intuition is there's a self-averaging in these random matrices. And the self-averaging comes from the fact that the standard deviation of the weights is scaled as 1 over square root of n. So for very large networks, the instantiation to instantiation to instantiation variability is also small. So if I have a smaller, you can also see that, if you just like generate a bunch of random matrices and you just look at the histogram, and you can see this.
Oh no, that I agree. I thought you meant after learning, the variability is smaller.
Also seems to be that way.
But then it means that if you make so I would have thought that you would have many solutions, many different solutions to which learning can take you. And so if your network is larger, you have even more different solutions because you have more parameters. So
it depends on the complexity again. So complexity seems to shrink the solution space.
Mm-hmm.
So the more complex the dynamics are that you want the network to fit, the smaller the solution space is.
Mm-hmm, mm-hmm.
And therefore, and this is something that I don't exactly know how to wrap my head around fully.
Mm-hmm.
So statement 1 is the more constraints there are, the smaller the solution space.
Mm-hmm.
Statement 2 is the more complex the thing is that I want the network to fit, the smaller the solution space is.
Mm-hmm.
These two I have observed myself and looked at it in many, many different circumstances, right? I haven't written down anything systematic about it, but here's the third statement. Which is immunity to the choice of the learning rule a good thing or a bad thing? Let's say I take a network and use target forcing like this to train it to get to a solution. Then I do backprop and get to the same solution. Is that a good thing? Or does it mean that it was inevitable and therefore stupid and nothing to do with biology?
Mm-hmm.
I don't know. I don't know the answer to this, and I don't think it's been clearly even articulated. The people that like universality, like claims for these types of networks, will say it's a good thing.
Mm-hmm.
I will say it's just the big, huge honking, Atlantic Ocean attractor that you just inevitable to fall into. Doesn't mean biology used it at all.
Mm-hmm.
And I don't know. I don't know at what point this breaks down into like philosophy.
Would you get the same variability in the solutions if you would start from a fully connected network?
So these are all fully connected, the ones I showed you.
Okay, yeah, they're fully connected, but from a Gaussian distribution. But if you would start everything from one, like fully connected to it and uniform.
So you won't be able to train these networks. Remember why you
they would, okay.
You can't train them because there's no dynamics to entrain. right? The dynamics come from the fact that you had this bulk of unstable eigenvalues that you're somehow making more regular by process of training. If you didn't have any spontaneous activity in these networks, then it becomes hard.
Yeah.
I don't know if there are other questions.
So maybe following the last question, it seems the network after training, they have some sparseness, right?
Also, I was hoping this, and answer no. So I thought, since everybody has a target function, you are this hyper constrained network, right? Like everybody has a target function. The network is like somewhere between 10,000 to 40,000 neurons big. And so then I was like, okay, if we're constraining everything, maybe the inter-area connections are automatically going to show up as sparse. That is not what happened. So now we're putting in sparseness constraint in the cost function. The first two papers was everybody was connected to everybody else. So we can tell directionality, but we can't tell anything about the sparseness. So now we put cost functions, like we put a sparseness constraint on the off diagonal blocks only, and we leave the within areas fully dense. And that's where we're finding some probability of connections between all of the blocks. And then we're doing this influence map experiment. When I say we, they are doing the influence map experiment, where I say, let's say Habenula and Raffi are connected by like 1%. Then they're doing this influence map experiment where they will take like 8 or somewhere between 1 and 32 Habenula neurons, and they will hyperactivate them only. And then they will image the whole brain and see if my predictions were right. Like they will derive this probability of connection from the influence map. But we didn't, the example I showed you today, no. I was hoping they would come out as sparse. That would be very cool.
So do you ever think of like initialize the network to have some sparseness? Like you initialize the network to be a sparse connectivity, and then you train the sparse network.
Sure.
So will this initialization lead to another result, or?
So you can do that and you freeze the zero weights, right? Like, you know, you take this random matrix that you have, and then you just say, okay, I'm going to take like 20% of those connections and set them to 0 and freeze them during training. Just don't apply Jij to all of them. Just apply J to some subset, the update, the delta term to the subset. And if the network is large enough, you'll still get a reservoir that's big enough.
Thank you.
You're welcome.
Any final 1 or 2 questions?
Really? Nobody wants to know what glia or cell types? Those are usually the brickbats I get.
I have at the other extreme, I have just a very speculative question, which is, can you speculate? So you showed us this nice example of application to the fish. Can you speculate a little bit, like, where is this field going? Like,
yeah, okay. Let me see. Am I tired and cynical, or am I upbeat and positive?
well, you can give both answers. I mean, I'm curious about both. Like, is it going to continue in like more and more complex artificial networks and you know, fitting, you know, using them as tools, and maybe also finding mathematical theories of why they are good tools and so on? Or do you think this is a transition to some other kind of description? I'm just curious what you think, how you think things will evolve.
Well, so let me just start by saying, I don't believe we'll ever have like this unifying theory of the brain, right? We're just going to have a pile of models. That's just how I think it's going to be. That said, the depressing view is when we take these tools super seriously. When we're starting to say things like, you know, the brain employs backprop, and I'm going to look for it and stuff. That's when it'll be a problem, because these are, you know, unrealistic kind of learning rules. It's one thing to say, I want to put cell types into this network because there are cell types in this brain region in the biological problem, or cell types are interesting because the eigenvalue distribution was different and I want to understand what that does to the dynamics, for example. But without a principled reason, putting stuff in is just stuff, right? That piece is the piece that worries me. And it has a, it's a little bit of the flavor of, just because you can record from everybody doesn't mean you should record from everybody. The analogy for us is just because you can build a 1 trillion parameter model and burn down rainforest doesn't mean you should, unless there's a principled reason for doing so in the biological problem. So that's a depressing view, right? We're gonna go into this inevitable, let's do more thing without thinking it through. But then there's the less depressing piece, which is, okay, we're like at this amazing confluence, right? Machine learning people are burning down rainforests and whatever, but they're actually innovating in this arena, right? If you can parse the developments from just the shit that appears in conferences, you'll be able to find amazing nuggets there, right? Like people inventing new types of learning rules, people looking at neuromorphic stuff and then stumbling into something that we can use and borrow. So that, combined with the fact that compute and storage is getting cheaper, combined with the fact that experimentalists are taking us super seriously and giving us data, actively seeking us out to give us data. So it's this kind of magical moment. And so if we have a chance to write down theories that go beyond mean field and really innovate, now's the chance. And I think we are doing it, right, to some extent, right? I do think that thinking about the geometries of these types of motifs is going to lead to some kind of principled way of looking at the collective. Large networks combined with large data sets and making sense of them in the simple way is important. Mm-hmm. So I think that's the less cynical view. So I don't know which of them is going to be right. Mm-hmm. But I think in the future, there's going to be a few elements that will be inevitable for us to tackle, like cell types, like neuromodulators, like connectomics, right? And I have come 180 on connectomics. I used to think it was the stupidest thing ever. This is all being recorded, by the way. It was the end of my career right here. But, I used to think, right, like, oh my God, these are dead brains and they're just weights and so what? But fast forward now 10 years later, and I'm like, ooh, this is an actual connectivity map. Like what happens if you initialize a network to this connectivity map? There's the hemibrain of the fly is solved. Can that generate the activity in the other hemibrain? It is useful. I'm glad I'm not doing it, but I'm glad it's there. That's going to become super key. Constraints from reality, cell types, neuromodulators. I am worried about glia, because they keep coming and going. Like every few years, people are like, ooh, glia and ooh, astrocytes, and then nobody talks about them for 10 years. So I don't know, but it's really, that's it. Those would be my bets for the future. What do you think, in fact?
I don't know if I have very developed thoughts on this, but now a question that will betray my ignorance is, has this kind of approach been applied to organisms where you could apply to all the neurons, like C. elegans?
Not very well, and I don't understand why. Well, for me, I can tell you why I haven't done it. I can't find a student or a postdoc interested in it. It's this weird, like driven by fashion's trend.
Strange, because there you have behavioral repertoires that are fairly well defined. You could have those as objectives and so on. So, I mean, that seems like a very natural.
Absolutely. Somebody should totally do it. Plus, the connectome is fully known.
Yeah, exactly.
Plus, people like Andy Leifer are imaging in these enduring behaviors. Yes, 100%. And 300 neurons are sufficient to do these things with. Somebody should.
Okay, strange.
Yeah, 100% should.
Okay.
Because if we have a chance to build a truly generative model, that would be the place.
Mm-hmm. Mm-hmm. Yeah.
But I think, yeah, the smallest organism that I can get my people to be super interested in is the larval zebrafish.
And how many neurons does it have?
100,000.
Mm.
See, but its connectome is getting kind of close to being solved. It can be, it does this kind of behavior too, right? Mm-hmm. It also has like social behaviors, like they school under threat in a variety of ways. They have amazing genetics in it, optomagic. So hence the natural interest towards the, ooh, look how pretty.
But the whole organism nervous recordings are with calcium imaging, right? So it's,
that's right.
The temporal resolution is not.
Oh, totally. This is really terrible temporal resolution, right? Like this nuclear localized GCAM, so rise time of about 1 second, sometimes 2 seconds, decay time of 5 to 7 seconds.
Yeah, so that
I mean, the monkey has done like 1,000 trials and gone home by this time.
Right. So that data is not very useful for you, I guess, or?
Which is?
The
data?
calcium data.
No, it is very. It doesn't matter, right? The RNN kind of doesn't care. That's the piece. It doesn't care about the resolution of the data. You give me spikes, we can do this. You give me ePhys, you can do this.
Oh, that's right.
You see, that's the advantage. So you get some notion of this, of the connectivity that gave rise to the dynamics. Then the question becomes about how seriously you take it, what you can do with it.
Okay. Any final questions? Well, if they're not, so let me thank you again, Kanaka. That was a very, very nice set of lectures and a really appropriate way, I think, to end the course. Thank you very, very much for all your time, for preparing all these lectures, for giving, you know, there was, by the way, we had a feedback session and many people were really excited by your lectures. So.
Oh, that's good to hear. Okay, that makes it worth my Saturday.
There were many people very happy with your lectures.
That makes me so happy because, you know, I don't teach in my day job. I don't know if you guys know this. I work in a medical school. Medical schools don't really have arts and sciences, so there's no like undergrad population to teach or anything. So this is like, I spent a week on this. So, you know, it's not like I just got up and then this, right? I spent a week preparing the lectures and.
No, I can tell, and I think everybody can tell, and we're grateful.
No, I'm happy. I'm happy for the chance. And the discussions have been really good. I mean, really brilliant questions.
Thank you so much. Thank you, everyone. I hope this school will have been useful. You can get something out of it. And thank you again, Kanaka. And see you, we'll be in touch with some of you, I'm sure. And I hope to see you again, Kanaka, in person.
Sounds good. I hope so. I hope so.
Okay, have a good evening. Bye-bye.